this is the title of my thesis, Statistical and Machine Learning Methods for Identifying Clusters of Variables with Applications in Omics, Ecology, and Psychology. All of that vocabulary will be explained in this presentation, so don't get scared away already. So here we go. Let me start off with describing to you the research problem that I looked at during my thesis. So some background to start off. The data that we analyze is often collected in a tabular format. So this is something like what you would see in an Excel sheet, where you'll have rows, which in this example here represent patients. And then we also have columns, such as these here, which measure the age, sex, blood pressure, body mass index of these patients. The columns of this Excel sheet are called variables, and so I'll use that vocabulary for the rest of the presentation. The rows are called observations because we observe these different variables for the patients. And then lastly, the data table is called a matrix. So those three vocabulary terms will come up quite a bit, so I just wanted to make sure we're on the same page. So the main subject of my thesis is clustering, which is a type of data analysis where objects are assigned to groups of clusters based on data. So this is a diagram demonstrating this idea. Basically, we take similar objects and we try to put them into the same cluster, whereas different objects are assigned to different clusters. The most common form of clustering is called observation clustering, and this is the clustering where the objects to be clustered are the observations. So in my example from earlier, this would be the patients. So given a set of variables, for example, the age, blood pressure, etc., what we would want to do is to assign these observations, the patients, to k homogeneous clusters. In the example that I show here, we basically have 4 clusters of patients. And the objective would be to find those clusters based on the data. So that's what we did here. We assigned these different patients to 4 clusters, one of 4 clusters. And then one interesting application of clustering observations is that we can use that to develop, for example, targeted treatments for patients. So if you consider that a certain disease has different subtypes of patients, if you can identify those different subtypes, then you can develop particular treatments for that particular subtype of patient. And that can be quite useful. So that was observation clustering, but the actual subject of my thesis is variable clustering, which is a little bit different. The idea here is that we want to assign the variables to clusters instead of the observations. So let me give you an example of a case where variable clustering would be interesting. This is an example from the field of transcriptomics where we deal with what's called gene expression data. So gene expression data basically tries to quantify the activity of genes in a cell. The observations in these data sets could be patients. So you might have a set of healthy patients and a set of sick patients. And then what you want to do is measure the gene expression for a set of genes. And the variables in this case are the genes. And those are the objects that we seek to cluster here. So the reason why this is an interesting thing to do is because we assume that for complex diseases such as cancer or heart disease, we assume that certain groups of genes actually work together to contribute to the development of disease. So that's what's shown in this diagram here. We have, this is a toy example. Obviously, there's far more genes than what I'm showing here, but there are 5 clusters basically of genes. 2 genes are connected or related to each other if they're connected by a black line here. We see that there are 2 clusters, the red cluster and the light blue cluster here, that have arrows that are pointing towards Y. So Y in this toy example could be the development of the disease, whether or not someone gets cancer, for example. And clearly these 2 groups of genes are both contributing to the development of disease. So identifying these groups can be pretty useful. Number one, because it allows us to better understand the mechanisms of disease, give us kind of a biological understanding. And based on that understanding, we can develop targeted treatments for patients where we really zero in on the genes and the processes that are important for stopping the disease from developing. So that was an application of variable clustering. And now I will describe the 3 main uses of variable clustering in the scientific literature. So the first and most obvious is that variable clustering can be used to identify grouping structure in variables. Often this grouping structure is unknown, so it's kind of an exploratory analysis to figure out which of the variables are related to each other. A second and really important use of variable clustering is called dimensionality reduction. So the idea here is that you start off with your Excel sheet and imagine that you have many columns and just a couple of rows here. Well, that's really hard to analyze for a data analyst. And so the idea is you would use variable clustering to kind of reduce the number of columns that you need to analyze. Here, what happens is we've clustered the different columns of our matrix of the different variables into 3 different clusters here, and then we take some kind of summary measure of the cluster, and we generate a new lower dimensional matrix here, which takes the main information from the really big data set and squeezes it into just 3 columns that are much more easy to analyze. And then finally, a third use of variable clustering is for variable selection. So for some of the clustering methods that exist, there's a setting so that certain variables can be assigned to none of the clusters at all. And so this essentially creates a kind of garbage cluster, a set of variables that are considered to be irrelevant and not useful for any type of analysis. And so this variable selection step is important because it allows us to distinguish the relevant variables from the irrelevant variables. There are 2 main approaches to variable clustering. The first is unsupervised, that's the word that we use for it. And what this means is that we only use the information from the variables themselves to perform clustering of the variables. So that's what we see here. The columns of this matrix here are each assigned a color, and variables with the same color are in the same cluster. A second approach is supervised. So the idea here is that we take another set of variables called the targets, and we're going to use them to help guide the clustering of the variables here. So it's adding extra information into the mix to supervise the clustering. Okay, so now I think you have all the background necessary to understand what the basic idea of the thesis is. So the idea here is that we wanted to develop new methods for variable clustering that allow us to gain insights about the data. In particular, we want to do 2 things. The first is we want to be able to identify the important variables, so which variables are relevant for the analysis or for predicting disease or something like that. And then another thing is we want to be able to provide the relative importance of variables within a cluster, because that makes it easier to perform further analyses. We know which of the variables to start studying first because we know that they're the most important in the cluster. We looked at variable clustering in a couple different data analysis settings, first in the unsupervised setting, and then next in the supervised setting, where we looked at the case where there's a single target variable and the case where there are multiple target variables. And all of these analyses were inspired by problems that are coming from the fields of omics, ecology, and psychology. So I just wanted to give you a kind of an overview of the different contributions during the thesis. All of the boxes in light blue indicate a method that we developed and implemented during the thesis project. And as you can see here, they fall under these different categories of unsupervised and supervised, etc. And most of these were presented in 3 main chapters in the thesis. Okay, so I feel like there's a lot of stuff that's going to be said during this talk, and I want to make sure that you guys are concentrated. So I added some memes to the slides so that you can have some like entertainment, right? So that your brains can relax. And speaking of brains, here's a nice one. Hope you enjoy. This one is like the story of my life. So this is you get to know me a little bit more while we go. Okay, so that was enough funny stuff. Okay, unsupervised variable clustering. So if you give a blood sample or a urine sample, that sample can be analyzed on a bunch of different biological levels. The first level could be in terms of the genes that are in it, and that's what's studied in the field of genomics. You could look at the messenger RNA, and that would be the field of transcriptomics, et cetera, et cetera. If you're looking at the metabolites, then you're in the field of metabolomics. So the field of metabolomics is the field that is interested in the fluctuation of metabolites, and that's what they analyze. So what is a metabolite? You can think of a metabolite as kind of a molecular fingerprint at a crime scene. So the crime scene is your body, unfortunately, and the crime would be some kind of biological process. So for example, the development of a disease. And the hypothesis in metabolomics is that if you can study the metabolites, you'll figure out that certain changes in metabolites might be able to explain or predict disease. So we can just look at fluctuations in metabolites to do those things. Question is, how do we quantify these metabolites? How do we take your data sample of urine or blood and turn it into an Excel spreadsheet? Well, one of the most popular ways of doing this is with nuclear magnetic resonance, or NMR spectroscopy. So this is turning your patient sample into a data matrix, which is what I show you here. This is just kind of a diagram of what it would look like, where we have our observations on the left and then our variables here that are the columns of the matrix again. So I wanted to show you what an example observation might look like. So this is a visualization of observation 4. So this is patient 4. And what I show you is each dot in the graph represents the value that's in the cell in the Excel spreadsheet. So for example, this first dot represents the intensity of the first variable, so the first cell for that row. And so I've plotted all the dots, so these are all the numbers that are in that row. The numbers correspond to intensities. And in practice, what we do when we visualize these spectra is to actually draw a line that's going to connect all the different points here. So this is what people normally look at, and it's called a spectrum. So this is another vocabulary word. By the end of this, you guys are going to have a lot of vocabulary under your belt. It'll be great. Okay, so let's talk about some of the properties of NMR data. Number 1. The molecules, so the metabolites that we're studying, and the ones that are present in the sample are represented by peaks at fixed locations in the spectrum. So in this example here, we have, for example, the molecule 1, which is the red molecule. It's all of these columns here that are in red. And this molecule 1 will always show up at this location in the spectrum. You can think of this as if the molecules each live at a certain address in the spectrum. And if you want to generate a new spectrum, they'll always be in the same spot. And then the variables that are at that address, so all the variables, for example, here 0 through 12 per se, all of those variables represent the same molecule, and so they essentially form a cluster of variables. Aha, clustering of variables is already coming back. Okay, second property of NMR spectrum is that the peak height, so the peaks that we see here, their height is proportional to the molecule concentration. So in this example here, we have 2 patients, and the relative concentrations of the molecules they've varied between the patients, and you can see that that's reflected in the height of the peaks in the spectra that are visualized here. So why is that important? Well, because this means that NMR data indirectly quantify molecule concentrations. So if you use NMR, this is a way of kind of capturing the different molecule levels in a sample. And that's what we wanted to do in the first place. Okay, so let me explain an example problem from Metabolomics. So imagine that we have a data set that has sick patients and healthy patients, and then we collect their samples, and then we use NMR spectroscopy to generate these spectra here. What we want to do is try to be able to predict which of the patients are sick and which patients are healthy just based on the spectra. So imagine that we know, in reality you do not know, but imagine that we know that the concentrations of molecules 1, 2, and 3 vary between patients. So that means that these molecule concentrations and the peaks in the spectrum are going to be different between patients. Well, those molecules can potentially be used to predict the health status of the patient. Potentially, I said, potentially. However, it turns out that only molecule 1 and 3 actually predict health. So those are the molecules that we want to find in the spectrum. And so what we need to do is find the spectral regions that are predictive of health status, and this means finding the molecule 1 and 3 in the spectrum. And once we've done that, we can use that area of the spectrum to predict the health status of new patients. So in order to predict health status, we need to be able to distinguish predictive regions of the spectrum from non-predictive regions. And that's a variable selection problem. We're trying to distinguish relevant from irrelevant variables. So that's what's shown here. We've identified molecules 1 and 3. All of those variables are potentially interesting for predicting. But if we want to understand the disease itself, that means that we need to identify the potentially predictive molecules, because we want to understand what is it about these molecules that makes us develop disease. And so in order to determine the molecules, we have to first perform variable clustering. So this is taking all of those relevant variables and splitting them into clusters here, where we have the cluster 1 on the left and the cluster 2 here on the right. And then after clustering, we can potentially associate those clusters with molecules themselves. I'm not going to talk about how that's done here, but know that you can do that, and this clustering is an important step for that. So why is this problem challenging? Well, you can think of finding these predictive clusters of variables it's kind of like searching for gold. Imagine that you have this spot of land here that, think of it as a spectrum, okay? And then we have three different things in it. We have dirt, we have pebbles, and then we have gold. And gold is what we want to find, right? So what do I mean by this? So dirt, in our case, these are the variables that don't vary much between patients. There's no way we could use those to predict whether someone is sick or healthy. Then we have pebbles, and these are variables that do vary between patients, so they could potentially be used to classify patients, but in reality, they don't predict health, so they're not useful. And then lastly, we have gold, and these are the variables that vary between patients, so they could be used to classify sick versus healthy, and they also happen to predict health, so they're actually useful to us. The problem is that gold and pebbles, they look very similar unless you have like an expert next to you that can help you distinguish the two of them. And lastly, there can be a lot of dirt. So that's not good. So the proposed solution that we suggested was to basically offer a way of pre-processing this data to make things easier. The first idea is basically remove all of the dirt that serves no purpose, so we have to get rid of it. And then secondly, the goal would be to identify packets or like stretches of gold and pebbles in the dirt so that you can put those aside and have an expert analyze those later. So that was a method we called adaptive clustering around latent variables, or ADA-CLV. The objective was variable clustering, selection, and dimensionality reduction all in one, which can be applied to spectroscopic data like NMR data. This is a publication that we did on that method. And I'm going to explain how ADA-CLV can be used in a pipeline for what's called biomarker discovery. So another vocabulary word. Biomarkers are biological variables that can be used to predict disease, development, or progression. So these are basically the relevant variables in a problem where you want to predict sick versus healthy, or whether someone's going to get sicker. So step one, we take our data matrix here, and this is a visualization of a spectrum just to remind you what's in that data. We apply ADA-CLV, and ADA-CLV does two things. The first is that it performs variable selection. So as you can see here, all of the columns that are in light gray, they have been unselected, as it were. They have been removed from the analysis and identified as dirt. And then the remaining variables have been clustered into clusters. So each color represents a different cluster. And so that these clustered here, this is the step where we're identifying packets or stretches in the spectrum that contain potential gold, so gold or pebbles. And then in the output of this, we have these selected clusters that are potential gold packets. We don't know which ones are gold yet, right? But we will next. So in the second step, we're going to try to look at what we found with ADA-CLV. We use what's called supervised learning, which is basically the equivalent of bringing in an expert who will evaluate whether the pebbles are gold or whether they're pebbles. And then afterwards, we have identified these biomarkers, and that's basically finding all of the gold packets that were in the dirt. So the general idea of ADA-CLV is that we assign variables to clusters using an adaptive threshold, hence the name ADA-CLV. This threshold is different for each of the clusters, so it really adapts to structure in the data. And all of the thresholds are continuously updated as the algorithm progresses. Some of the variables will be assigned to multiple clusters, so this is useful because it means that we can have clusters that potentially overlap. And then some of the variables are assigned to none of the clusters, and that means that those variables are identified as irrelevant. These are the dirt variables, and that's a variable selection step. And then another thing about ADA-CLV is that it's actually going to calculate cluster membership degrees. So rather than saying, okay, this variable belongs to a cluster or it does not, we say to what degree it belongs to that cluster. So I visualized what that looks like here. So each of these panels represents a cluster. And then the x-axis here is the variables, and then the y-axis is the cluster memberships. And you can see that we kind of have these peak-shaped cluster membership degrees, where, for example, in this first cluster, the top, this variable here has the highest degree of membership. So this is the probably most important variable in the cluster. And then this type of visualization can also be represented like this. And so I'm going to use this format for the rest of it. But as you can tell here, each line represents a color. Each color represents a different cluster. And I just wanted to show you some results of how this method works. Here we're going to be comparing a case where we have lots of data, which is the good case, and another case where we have not as much data, and that would be the less ideal case. On the top, we have the true cluster memberships, and on the bottom, we have the memberships that are estimated by Ada Silvi. And as you can tell, they're quite similar, right? So we're doing a really good job of recovering stuff. And the only difference perhaps between these two cases is that less cluster overlap is detected when we don't have a lot of data. So in conclusion, basically, we can detect this cluster overlap if we have lots of data. And then in the second case, I'm comparing the case where we have the correct number of initial clusters provided to the algorithm and the case where we have too many initial clusters. And again, we have results that are quite comparable to the truth. So even when we have too many initial clusters that are provided, we're able to recover the true clusters. Okay, so it seems great, right? Like it's a great method, I think. But there are obviously limitations, right? So one of them is that it's good for data. So it's good for data exploration, identification of potential biomarkers. But the limitations are that sometimes it finds these redundant clusters. So it'll find the same cluster multiple times. And in theory, that's not really that much of a problem, but it is a little bit complicated to merge the clusters afterwards. It adds an additional step that it's kind of a pain, right? But I mean, it's fine, but it could be better, right? And then the other thing is that the variable clustering and the variable selection step is totally unsupervised. We kind of ignore that there's a gold expert somewhere. We don't even invite him to the process at all. But an alternative would be to include that gold expert from the beginning so that the process is supervised. And that would avoid us from assigning gold, pebbles, and dirt to the same cluster, which is undesirable. Okay, so it's going to be continued, but first we have to take a break for our brains. So another meme break. So this one's based on a true story, like all of my thesis. So no, it's fine. We got there eventually. Okay, so the next chapter is on supervised variable clustering. So now, in addition to performing variable clustering and variable selection, we have an additional objective, which is to predict target Y based on the variables in our data set. So for example, Y could be the health status of a patient. Is the patient healthy or sick? And in this chapter, we have this variable clustering method that's really built for this prediction problem. So the context that motivated this was from the field of transcriptomics. I explained this at the beginning of the talk, but as a reminder, basically the idea is we want to find groups of variables that predict the health status, and in this case, the variables are genes. And the objective is prediction of the target, so the health status of the patient, the blood pressure, some kind of variable like this. We also want to perform variable selection, which means we want to identify the genes that are relevant for predicting the target. And then lastly, we want to, of course, perform variable clustering because we want to identify this group structure that is actually unknown currently. So what are the challenges for data that has cluster structure? Well, one of the problems is that for the variable selection problem, which is distinguishing irrelevant from relevant variables, methods tend to over-select variables. So they tend to say that many variables are relevant when in fact there's not quite as many that are actually relevant. And then a second problem is that in terms of prediction, when there's cluster structure in the data, the prediction accuracy tends to decrease. And so the solution that's proposed in the scientific literature is to use a type of modeling that directly accounts for cluster structure. There are two types of methods. The first method is called embedded methods. These methods are simultaneous, so they simultaneously build a model for prediction and perform clustering. And then the second type of method is two steps. So it performs clustering first, and then afterwards performs the modeling. Now, these two types of approaches have advantages and disadvantages. For example, the embedded methods have the advantage of supervising the clustering, which means that they're bringing in that gold expert from the beginning, which is quite advantageous. They also perform variable selection during clustering, so they're removing the dirt right away, which is also good. The two-step methods have the advantage of being computationally efficient, which can be a real advantage if you don't want to wait a really long time for your results. And then the second advantage is that they're more easily modulable. You can apply them to a lot of different types of modeling, and so they're more flexible. But obviously there's disadvantages to both sides, right? So our question was, is there some kind of better way to do this? And our proposition is to improve that, and it's called variable cluster principal component regression. And this is a method that we're going to present in a paper that we'll submit at the end of the month, hopefully. Okay, so variable cluster principal component regression. So the first step here is to perform supervised variable clustering and variable selection with a method we call weighted SOS NMF. You can forget that, it's not that important. Anyway, so the idea is we bring in a variable here, the target variable, that's going to help us out when we're assigning variables to clusters. Notice that here in this diagram, I have some of these variables that have a white color. These white variables have been removed. We've considered them to be irrelevant. And so we've already performed this like variable selection step. And then the remaining variables are assigned to clusters, which are indicated with the colors. In addition to the cluster assignments, we also do those cluster membership degrees that we had with AdaCLB. And this is important because it gives the practitioner kind of guidelines for which are the variables to investigate first when they're trying to validate the biomarkers that they find. The key contribution of this first step is that we've added supervision because there really aren't any methods that have this supervised clustering step like this. And then in the second step, we calculate what's called a latent variable based on the clusters from step 1. So I'll explain what that is. Basically, we use the cluster assignments. Here I have an image here of the different clusters, and then we also have this set of variables that were removed that were considered to be irrelevant. And basically, we calculate some kind of summary measure of the cluster, and then we gather it into this new matrix. And so these are called latent variables because they're supposed to summarize latent information that's kind of unobserved, but is hidden in the data. And then once the latent variables have been calculated, we use those to learn a model for predicting the target. And then once that model has been learned, what we can do is provide a new patient for the model, and then we can use the model to identify whether that patient is sick or healthy. So this slide, the purpose of this slide is just to show you that the method we propose performs better than the other methods. So this is just in terms of the identification of clusters. What I'm showing is 3 simulation configurations that we tested, where we have the number of variables selected, which is in the bottom, and then on the y-axis, we're looking at the quality of the clustering. And so the vertical bar here at about 20 variables, that's the true number of variables that we should be finding. And when you look at that, you see that our method, which is this dark red method here at the very top, it achieves a better clustering performance than the other methods for pretty much any model size, but especially for the true model size, which is 20. So the takeaway from this slide is just that we perform better for clustering. If that's all you got from it, that's great. Okay, another meme break. This is a good one to remember because sometimes we really want to like do magical things with our methods, and you got to be realistic about it. So life lessons. Okay, so now the last important chapter was on supervised variable clustering when we have multiple targets. So in the previous chapters, the purpose of clustering was really just to understand the structure in the data X. In this chapter, we're going to use clustering for a slightly different purpose. The purpose here is to assign meaning to multiple targets Y. So to better explain that, I have this diagram here. In the first case, in previous chapters, we had a single target Y, which is this column here, that is associated with multiple clusters of variables. So you see here we have a green cluster that's associated with Y, we have a red cluster and a blue cluster, all of them with the same target. And in this chapter, each target is going to have a single cluster of variables. So in this example, the green cluster is only associated with the first target. The red cluster is only associated with the second target. Okay, so I'll go over an example problem from psychology, which was what motivated this chapter here. So imagine you have the following research question. What are the major attributes people use to judge the similarity and difference between social groups? So for example, say you have the two social groups, artists and conservatives. The researcher is interested in understanding why these two social groups are seen as different by people. It's not objectively are they different, why are they perceived as different by human beings. And so one research strategy you could have would be to just ask the participants directly, hey guys, why do you think these two social groups are different? Unfortunately, this is kind of an unreliable approach, right? Not only are people a little bit biased, but it's not always like super helpful to just ask them directly. Another better strategy would be to analyze the data to uncover what the implicit perceptions that people have about social groups are. And this is definitely a more objective approach. So to do this, we have to collect some data. And so the data setup is the following. We ask participants to rate the similarity of pairs of social groups on a scale from 1 to 9, where 1 indicates that the groups are very similar and 9 indicates that they're very dissimilar. So in an example here, we could have the comparison between students and educated people, where we give them perhaps a score of 2, because they're mostly very, very similar. And then we have students and conservatives, where we give them a score of 6, because they're mostly different, but they're not too different, so somewhere kind of in between. And these different scores will be collected in, once again, an Excel spreadsheet, which looks like this, where on the rows we have the social groups, and in the columns we have the social groups. And so every pair of social group is compared and the scores are collected in the data set. The problem with this data is that the data has way too many columns for us to really analyze. And so one solution to that problem is to perform dimensionality reduction. And so we take our matrix that has 80 rows and 80 columns, and we try to extract just the most important information from that data set and summarize that information in these 2 columns here. And why 2 columns? Well, the advantage of 2 columns is that now I can visualize my data using a 2D plot, which is helpful. We want to learn things about the data, so we need to look at it. So in psychology, multidimensional scaling, or MDS, is often used as a method to perform this dimensionality reduction step. And the new lower dimensional data is called an embedding. So that's, again, another vocabulary word that you'll need to remember. This embedding here is the kind of the summary of the major information in the data set. And when we have just 2 columns, then we can visualize that with the 2D scatter plot. So what you see here, if you see, for example, the point here, which is for homeless people, this one's really far away from Republicans, for example. And so that means that in the high dimensional data set, people perceive these 2 social groups to be very different. Whereas you have, let's see, what else do we have? We have hipsters and hippies that are right next to each other. And so that indicates that people thought that those 2 social groups are quite similar. The problem with these MDS embeddings is that we don't understand why the MDS places some of these points closer together and some of them far away. I mean, we can make hypotheses, right? But scientifically, we don't have like an objective explanation for why the MDS did that, because the axes, so the dimension 1 here and dimension 2, they have no predefined meaning. So why is it important for the axes to have meaning? I think it's easier if I give you an example that shows you the difference between the 2 situations. So in the first setting here is the traditional setting where the axes have meaning. Basically, I've taken 2 variables and I plot one variable with respect to the other variable. In this case, a variable that is the scale from traditional to modern, and another variable that is the scale from masculine to feminine. So when I look at this plot here, if I ask myself the question, well, hey, why is it that men are all the way to the left here and women are all the way to the right? Well, it's because this dimension, the horizontal dimension, is the scale from masculine to feminine. So it's very easy to explain why the points are distributed as they are. With an MDS embedding, the axes have unknown meaning. So if I ask myself the question, hey, why are teenagers and conservatives on opposite ends of the first dimension? I don't know why. There's no name given to this dimension, so I don't know what it means. So the question is, can we associate each axis in the MDS plot with an attribute? And if we can do that, that would be great, because it means that we can give meaning to the embedding dimensions, and that meaning can be used to interpret the position of points in space. So to do that, we need a second data set. In this second data set, the social groups are scored according to different attributes, in this case, stereotypes. So we have scores ranging from 1 to 100, where 1 is for the first stereotype and 100 is for the second stereotype. So an example here, imagine you have the social group artists, and we're on the scale from masculine to feminine. Well, maybe you'll say they're a score of 65 because artists are maybe a little bit more feminine. I don't know if that's my personal opinion, but that's what people said. Okay, and then the next thing is conservatives on the scale from traditional to modern. Maybe you give them a score of 17 because you consider they're more towards traditional than modern. All of that data is collected again into a data table here, or matrix, where we have the rows, which are the social groups, and then on the columns we have our different variables. Here are the scales between the different stereotypes. And so our objective would be to identify clusters of these attributes, so these stereotypes, that can be used to explain each of the dimensions of the MDS embedding. So here I'm just going to summarize what we talked about, so you have it in your head. So here the idea is we collected a first data matrix called Z here that collects similarity scores between all pairs of social groups, and then we use dimensionality reduction in a first step. MDS in particular, to generate what's called an embedding, which is a lower dimensional matrix, which is easier to analyze. But unfortunately, this embedding, the dimensions or the columns don't have a known meaning. And so what we want to do is use the second matrix that contains variables and associate clusters of variables with each of the dimensions so that we can understand things. So a potential strategy for this second step, which is associating variables with dimensions, is to use what's called sparse regression modeling. And that allows us to identify the important variables for predicting each of the dimensions of the embedding. So this is essentially a variable clustering and variable selection problem. Again, you have this nice diagram here where we see some of the variables are selected, those are the colored ones, and then we assign the variables to each of the dimensions. And variables that are selected for the same dimension are considered to be in the same cluster because they represent the same trends or the same ideas. The problem is that the rotation of MDS embeddings is entirely arbitrary. So this is an example of what the MDS embedding is that's given to you by the computer, but you could also consider this embedding here, which is a 99-degree rotation. And as you can see, the distances, the pairwise distances between all points remains the same. We're just rotating, right? And so in fact, both of these embeddings are equally faithful representations of the original data set. So they're both equally valid. But the problem is when we're using our sparse regression modeling technique, what we find is that the error and the interpretability of the sparse regression models is not equal for both of the rotations. So the rotation actually does matter. And so here's an example to really emphasize this point where I show you two cases. One where the model has more error and is less interpretable, and the second one where there's less error and it's more interpretable. So in this first example, the 93-degree rotation, we require basically 9 variables to explain the dimensions. So you have to keep in your mind 9 variables all at once to understand this. Also, the error is relative error of 0.38. And then if we rotate that just by 6 degrees, so if we're really just moving a tiny little bit, we find a model that has less relative error, so it's predicting better than the other one. And it only requires 4 variables to explain the dimensions. So this second rotation is much better for us. It's much more faithful to the prediction process. And then also for our poor little minds, we can actually understand because there's only 4 variables to keep in mind at once. So this is definitely the preferred rotation. So this is just to suggest that finding the rotation, the best rotation for the embedding, is actually a very important problem. And we need to find the rotation that allows us to really balance the low error with the high interpretability. Okay, so we proposed 2 solutions called VIR and BioT, which were presented in these papers. The main idea is that given an MDS embedding and a set of variables, we want to find the best transformation of the embedding. So for example, a rotation of the embedding, and then also find a linear model that can be used to explain the embedding. And we want to do that in such a way that there's low error and low model complexity. So we want as few variables as possible to be in the model. And this balance between error and complexity, the relative importance, that's something that can be defined by the user. So it's quite flexible in that way. So for the two proposed solution, beer can only be used for rotations, and it's only applicable to embeddings of 2 dimensions. And then BioT is an improvement of beer that essentially finds any orthogonal transformation, and then it can be applied to dimensions or embeddings of any number of dimensions. So you don't even need to visualize the data to learn something about it using BioT. So let's go back to our research question. We wanted to know what are the major attributes people use to judge the similarity or differences between social groups. And now I'm going to show you what BioT proposes as an answer to that question. Here I'm looking at an MDS with an embedding with 3 dimensions, and I'm going to show you 2 dimensions at a time. So this is the dimensions 1 and 2. The arrows in the plot represent the variables that are used to explain the dimensions. So for example, the first dimension, which is the horizontal dimension, the major variable that explains this dimension is the poor wealthy trend. And that makes sense because when we look at it, we see homeless people, poor people, lower class people are on the left. And then on the right, we find white collar, upper class lawyers, business people and stuff. So that's the major variable, but there's also some other variables in that cluster. We have the light purple variable, so the religious scientific trend, and then also the uniformity and diversity trend that can also be used. So those first 3 arrows are the major variables in the cluster that explains that dimension. And then for the second dimension, which is the vertical one here, we have 3 major variables in that cluster. The first is traditional modern, then we have conventional alternative, and then finally loyalty and autonomy. So to summarize, people in the top here, so we have hipsters, goths, atheists, hippies, bisexual, all of those people are perceived as more modern, alternative, and autonomous, whereas people on the bottom here, we have elderly, old, Christians, religious, Muslims, they are perceived as more traditional, conventional, and loyal. So those were the first 2 dimensions, and then for the third dimension, we can look at this plot here where the third dimension is plotted in the vertical axis here. We again have a cluster of variables that can explain that. So the major variable explaining this dimension is the smart and not smart dimension. And apparently women are considered to be more on the smart section. So who knew? People thought that. I don't necessarily believe that, but people perceive that to be true. And so we have Buddhists and Hindus that are considered to be more smart, more altruistic, and more feminine, whereas we have criminals, rednecks, athletes, jocks, sports fans, and military people who are considered to be less smart, more egoistic, more masculine, and then also more competitive. So yeah, so this was just to show you that basically we can use BioT to understand why the MDS is placing certain points together, which in a sense allows us to understand why people perceive certain groups to be similar or dissimilar. This guy, he's like, when is this presentation going to end? Well, it's almost over. Okay, so now I'll just talk about the conclusion and future works. So to summarize, this thesis proposed methods for analyzing data, and these analyses involved variable clustering. We looked at unsupervised variable clustering, where we don't consult our gold expert at all. And then we also considered supervised variable clustering, where we consult our expert from the beginning. We looked at cases with a single target and then cases with multiple targets. And then for future work, well, is there anything left to study? Well, all of the methods presented here, they depend on certain user settings. So examples are the number of initial clusters that are provided, the degree to which we accept a complex model. So how many variables are we allowed to put in the model? All of these things must be defined by the user. And actually, currently, it's quite unclear how you're supposed to choose these user settings, especially if you want to simultaneously optimize prediction, variable clustering, and variable selection. It's really the simultaneous optimization that's quite difficult. So I have a couple ideas for that. Obviously, my thesis is over, but maybe the next doctoral student will take this up. One thing you could do is try to kind of invent some kind of numerical criterion that summarizes all of the qualities at once. And then you just need to optimize this criterion to try to optimize all 3 things. And another strategy would be to change the method so that the best settings for one of the qualities, for example, the best settings for prediction also coincide with the best settings for variable selection and clustering. And if you can get your method to work like that, well, then that's great, because then you really only need to optimize for prediction, and then necessarily you will also have the best settings for variable clustering and selection. So this is the summary of all of the different papers that I completed during my thesis. And thank you very much for listening.